the lecture of effect of mutation at different level. Today I will talk about the effect on DNA sequence level as well as in gene level. I will talk about the level at gene as well as DNA sequence and their effect on biological, genetic and evolutionary causes. I will talk about different other types of DNA mutation as well. Now, when the non-lethal mutations are getting accumulated, they gives different types of genetic variation. Majority of the mutation does not have any contribution towards the fitness. So, neutral mutation does not contribute to the fitness, but beneficial mutation has contribution to the adaptive fitness because they have reproductive success. Now, being permanent a mutation, that is actually repaired by the DNA repair mechanism, but if they are escaped from the DNA repair mechanism, the mutation becomes permanent. Mutation causes. So, as we know that for molecular decay, spontaneous mutation caused by DNA polymerase proofreading inaccuracy as well as translational DNA repair and other effects of the DNA repair may give rise to the spontaneous DNA mutation. Apart from which, there are some reasons like X-ray, chemical mutagen as well. They are acting as mutagen, physical or chemical mutagen causing the generation of mutation. Now, these mutation may cause at the different level like gene, genome, chromosomal, cellular, tissue, organ and different aspect of different levels. Here in this lecture, I will concentrate only at the DNA sequence level and gene level. Gene mutation. So, sudden and abrupt heritable change in DNA sequence is called as gene mutation. It gives the formation of new allele by genetic variation. So, new allele is better evolved and better adopted in the new environment. Different types of the mutation. Random mutation. So, mutation rate can only be increased, but according to the random mutation theory, the direction of mutation cannot be changed as the selection pressure of the environment is getting changed. But adaptive mutation theory states that by John Kearns in 1988, it was proposed that only when strong selection pressure is present, then only mutation causes the change in phenotype. Now, let us see about the pre and post adaptive mutation theory. According to Lamarckian theory, post-adaptive theory states that only when the selective pressure is present or selection pressure is present due to mutation, phenotypic changes happen. But the contradictory theory is the pre-adaptive theory states that randomly mutation happens and this random mutation is chosen by the selection pressure to evolve better adopted phenotype. And it was actually proved by Liederberg after 10 years of experiment with Luria and Delbruck in 1943, who has done their experiments with the T1 fudge resistant E. coli, and they have actually examined the variants in the mutants. And after that, Liederberg actually with some antibiotic resistance in replica plating technique proved the pre-adaptive mutation random theory. Now, paramutation. In 1950, Brink in case of Z. Mays have proposed the theory of the kernel color alteration by the effect, the heritable changes of other gene. Hypermutation. It is an acquired type of mutation and it is non-heritable. It actually done in immunological cell for better adaptation at the B cell receptor. So, continuously somatic hypermutation give rise to the differential changes of the B cell receptor, so it can combat against different new type of foreign antigens. mutated genes. So some of the genes having very higher frequency, and these higher frequency mutation rate is dependent upon the physiological factor, genetic factors, sexes. According to them, they are getting differentiated. But it was found in case of Drosophila, by omission at the portion of second chromosome, the mutation rate is getting decreased. So at the second chromosome, some mutated gene is present which impose higher amount of mutation rate towards the other genetic loci. And it was found that in case of E. coli, the Paul B locus is actually related to the transversion and prefer mutated genes, otherwise they can give rise to the transversion as well. So mutated genes can evoke more mutation rate. Number of genes from the mutation rate estimates. If big or capital U is the mutation rate of the total genome, n number of the mutation rate is getting happen in n number of genes. So if small u is the rate of mutation of each gene, then capital U is equal to capital N into small u. So capital U divided by small u is equal to the number of gene that is getting mutated. From this estimation of the mutation rate, number of gene can be easily calculated. Mutation rate. As I have already discussed, mutation rates are distinguished at different chromosome located genes. As we know that 0.1% related to the X chromosome per thousand, then it is increased in case of second and third chromosome for the Drosophila having equal amount of the larger size, that is equal to the 5 times larger than 0.5%. But in case of dot chromosome, fourth chromosome, it is much more lesser. In case of diploid organism in human, it actually accounts for the 2 gametes. As we know that the greater number of the mutations happen in case of virus than that of the bacteria. Now these DNA sequence means maybe single nucleotide giving rise to single base pair change, or maybe less than 1000 base pair. It may be point substitution or point source, or maybe non-point source. Now in case of point mutation, it may be the substitution or insertion deletion, and in case of non-point mutation, it may be the insertion deletion as well as inversion. Next one, single base mutation. Either it may be the substitution, that is changed of one nucleotide or base pair by another, or maybe insertion of one nucleotide or deletion of one nucleotide, which may give rise to the frame shift mutation. Now this point or single base substitution can be the transition or transversion. Transition means the change of purine by another purine, or change of pyrimidine by another pyrimidine. But in case of transversion, change of one purine by another pyrimidine, or change of pyrimidine by purine. So mutation at the level of gene. So level of gene means the stretches of the DNA sequence as well, and we can find them in case of insertion and deletion. The frame shift mutation may be happen. So when we are talking about the effect at the DNA sequence stretches or gene, that may be at the protein coding region or non-coding region of the protein. First, now point mutation or substitution, that may have various types of effect. So various types of effect means it may be at the protein coding region, changing the amino acid by means of missense mutation that I will discuss, either, or maybe at the intron level where it does not affect, or maybe at some sequence within the protein structure that may not alter the protein activity, or sometimes maybe at the repetitive sequence. Even it can be compensated by the non-mutated allele as well. Now effects of the coding region means, as I have already told, that substitution may bring the synonymous and non-synonymous substitution or result at the protein level at the amino acid. So in case of synonymous change, may be the neutral or silent, and non-synonymous change may include the missense and nonsense mutation. The first one, synonymous change, silent mutation. So here we can find the same type of the amino acid may come. So amino acid codon may not be changed by mutation at the intron region either, or at the degenerate portion of the codon. So here we see TGT is getting changed to TGC, T is changed by C, coding for cysteine both. Next, synonymous neutral. Actually, neutral is a type of missense mutation, but that means that does not change the sense of the amino acid. Suppose here glycine is getting substituted by alanine by changing CCA to CGA, change of C to G. So the meaning or the sense of amino acid does not change the activity of the protein as well, but it is the hidden source of variation. Next one, non-synonymous change. So non-synonymous changes happen in missense mutation, where different types of the amino acid is getting coded. Here we can find TGT is getting changed by TGG, T for G coding first for cysteine, then for tryptophan. Different sense of amino acid change can change protein activity. That may be non-conservative, where the R group, suppose initially it was coding for the polar amino acid, now it is coding for the non-polar one. Like glutamic acid is, if it is getting changed to the valine, like sickle cell anemia. So that cause the non-conservative missense mutation. But conservative substitution give rise to the fact the R group is changing for the same sense. That means if R group is changing from the valine to leucine, so it does not change more to the protein or enzymatic activity or amino acid sequence of the primary level. Now non-synonymous change at the nonsense mutation. Here we can find the protein coding codon for the amino acid is getting changed to the stop codon. Here we can find TGT coding for cysteine is getting changed to TGA for stop codon. So T is getting changed to A. So we can have the premature protein synthesis, so activity is abolished. Next one. Now effect of both missense as well as nonsense mutation. If nonsense mutation by missense mutation is read through, then it will give the long extension as the C terminus. So that can actually abolish the protein function. If the short extension happens, that does not change too much, but if long extension happen, it actually changes the structure and function of the protein. Next one, frame shift mutation, insertion and deletion. They can change the open reading frame by the insertion or deletion of one or two nucleotides. Now we can see if it happens in 3 of the nucleotides or multiple of 3, it can change the number of codon. So we can miss 1 or 2 amino acids in the final protein. Now effect of frame shift mutation can be happen at the ultimate protein, and we can find at the C terminus level of the protein is getting changed. The operator and repressor constitutive mutation with respect to the lac operon. If we find any sort of changes happen in the operator sequence, the sequence is unable to bind with the repressor protein, then the protein expression will constitutively turn on. So it will not inhibit RNA polymerase anyhow. The same thing in case of repressor constitutive mutation, the repressor protein is unable to bind to the operator sequence, again constitutively the protein expression will turn on. Now other regulatory mutation includes the reverse mutation, when by back mutation we can again gain back the original phenotype from the mutant phenotype. Now trans and cis dominant mutation. So trans dominant mutation is related to the diffusible protein, but in case of cis dominant mutation related to the operator sequence itself. In case of transposition mutation, the mutation happens by the insertion of the transposon. Other regulatory site mutation includes the splicing site mutation, happens at the intron splicing GTAG signal. So two introns are not well removed to form the particular mRNA structure from the primary transcript. That is happen in case of beta globin gene for beta thalassemia. And it can also be happen in the polyadenylation site, because we know that at primary transcript there is a signal for the removal and addition at the 3' end of the poly-A tail. If the proper signal is not removed, then long poly-A tail will be added, give the instability to the mRNA. Next, effects of frame shift mutation at the non-coding region may influence the regulatory site, promoter site as well as DNA protein binding site. So it inhibits the nucleosome assembly site as well and replication starting site. and we can find it can inhibit RNA protein and RNA interaction during the spliceosome and splicing sites. Direction of gene mutation, it may be forward, it may be reverse including back and suppressor mutation, which may be intergenic or intragenic. The next one, forward mutation. Forward mutation means from wild type we are having the mutant type, and in case of reverse mutation we are gaining back to the original phenotype, and this may be the true back mutation. At the same site we are having the mutation to gain the original phenotype, or maybe the suppressor mutation. So, suppressor mutation means at second site which helps to gain the phenotype, but original sequence cannot be gained, which happens in case of back mutation. So, it may be intragenic at same codon but different nucleotide, or maybe intergenic at the amino acid tRNA synthetase gene may bring the same amino acid for different codon. Now, mutation at the level of different aspects. So, it includes gene mutation. So, in cryptic manner gene is getting mutated, or genome mutation at the structural level of chromosome as well as functional level. And we can see the structural level is called the chromosome aberration. It includes translocation, insertion, inversion. Cytologically, gene mutation regarding this cannot be observed, but submicroscopically can also be observed, and segregation is heritable, and we can also find the reversion as well. In case of numerical chromosomal change, gene mutation also may be happened. Extranuclear mutation. So, at different aspects we are discussing about three different types: gene mutation, genome mutation related to the chromosomal aspect which contains the gene to be mutated, as well as extranuclear mutation. The extranuclear mutation obviously genetically inherited, but inheritance happens without the effect of nuclear function. It is inherited by cytosol. It is inherited by cytoplasm as well. So, we can see that happens in case of Mirabilis, Anthera, Drosophila, etc. Again, it may be of two types, plasmon and plastidome. Plastidome is related to the plastids, and that actually helps in sex differentiation, that helps in case of development as well, that helps in case of plastid development. Now, genetic status of gene mutation. Now, when we are talking about the genetic status, it includes the fact it may change in the amino acid sequence. Suppose from glutamic acid, the valine is getting intruded, which can abolish the protein structure function, as well as the altered sex ratio is related to the phenotype, which was found by CLB mutation in Drosophila. From homogametic sex, we are having the heterogametic sex at the 2:1 changed sex ratio of Drosophila. The phenotypic effect of gene mutation. It may be either absolute or may be the conditional. Now, absolute mutation means mutation at all condition, but the conditional mutation means only the mutation may be happened at some restrictive condition, environmental condition, and cannot be happening at the permissive environmental condition. Sometimes it may be related to the conditional lethal, just to destroy the organism, and it is two types, temperature sensitive as well as suppressor sensitive, and different other types related to the lethal and leaky mutation. The first one, temperature and suppressor sensitive mutation. In case of Zymes coat color, we can find the melanin synthesis gene or enzyme is not produced by the elevated temperature, but in case of the lower temperature, they are able to produce the melanin, so patches are observed at the claws only. The suppressor sensitive mutation is found by the effect of the suppressor at different sites, which help to gain the original phenotype. The lethal mutation actually reduce the potency of the longevity. It may be now or maybe after long times, or maybe sometimes a leaky mutation that reduce the growth in case of bacteria as well. Pleiotropy means the change in one particular genetic loci can control various phenotypes, like in case of HBS and HBA. In case of sickle cell trait, these two types of heterozygous hemoglobin may give rise to the malaria resistance, but it is the hemolytic anemia condition which at the homozygous condition give the sickle cell anemia as well as death. Mutation is used in the research because in mutagenesis with X-ray and other chemical mutagen, new trait can be evolved, as well as by transposon with model organism like Arabidopsis thaliana and Drosophila, the mutation can be introduced for experimental purposes. And tools of mutation include the mutant. Now here we will discuss about the detecting mutants. It may be the visible mutant like Drosophila eye color, nutritional mutant like auxotrope or prototrope in case of bacteria. Auxotrope requires some vitamins, some nutritional requirement, but prototrope does not. In conditional mutant, by lacking of DNA RNA polymerase with the gene knock treatments, and resistant mutants in case of E. coli can grown for the antibiotic resistance. Nomenclature: if Lu plus phenotype is observed at heterozygous condition, so Lu plus Lu minus heterozygous condition, Lu plus is dominant one, which actually give rise to the Lu plus prototrope can grow without the help of leucine is getting supplemented in the minimal media. Now mutant can be classified as the morphological mutant, which can only in outward phenotype, or biochemical mutant, the effect at the lesion of any enzyme related to the metabolic pathway. Morphological mutant, that is change in seed color, ultimately is the biochemical mutation manifestation. Mutation may be absolutely defective at all the condition, may be, or conditional, that is permissible for some permissive condition and restricted at the restrictive condition. And genetic analysis can be done by complementation as well as recombination. So recombination help to know the genetic map of the different loci at the chromosome, and complementation actually help to know whether it is regulatory site or not, in a particular phenotype how many mutants or how many mutations are getting happen. So use of mutation for the genetic analysis is used for detection of gene recombination frequency, because recombination frequency is directly proportional to the distance of the genetic loci over the chromosome. So help to map the chromosome. So we have discussed about different types of DNA sequence and gene mutation. There are different aspects to show the different types of phenotypes. Thank you.